So today we have two really great speakers. Our first speaker is going to be presenting her poster on different ways to treat cancer. So her name is Melanie, and she's a graduate student in biomedical engineering. And then we'll have a talk by Nick Fisk, who's a computational biologist who's studying evolution and human health, also somewhat related to cancer, and how we can use bioinformatics in different ways.
All right. Let's see. Hi, everyone. Can everyone see the screen? Thumbs up, maybe. Okay. Wonderful. Great. Yeah, thank you for the introduction. My name is Melanie. I'm a graduate student at Yale. I study a field called biomedical engineering, which basically what that means is we take what we know about biology and medicine, and we use that to design and engineer new ways to care for patients who are sick or injured or need some kind of medical care. So today I'm going to talk about a problem that many scientists around the world are trying to solve, and that is how to treat or cure cancer. So there are a lot of different ways that people are trying to approach this problem. And today I'm going to talk about a method that we're working on, which actually uses DNA to kill cancer cells. So you may be familiar with DNA when you've been talking about things like ancestry or genetics or genes. But today I'm going to talk about it and how we can use it as a tool for treating cancer. So I'm going to walk you through my science poster that I have here and how we design the experiment and think about the problem and come up with some of our next ideas to try. So I'll zoom in a little bit on each section as we go through, and please feel free to put any questions that you have in the chat, and I will do my best to answer them as they come up. So before I get into my experiments, I just want to go in a little bit more detail about what the problem is. So our bodies are made up of cells, trillions of cells. Cancer happens when a group of cells just starts to grow out of control. They multiply and they multiply. They can push out other healthy cells in your body. They can steal the nutrients that the other cells in your body need to survive. They're greedy cells. And this is what we call cancer. So I have here in this kind of diagram showing in green a row of healthy cells that are doing some function in our body that's important for us to live and be healthy. And then in red here, a group of cancer cells that are forming what's called a tumor and kind of pushing their way through the healthy cells and ultimately making us very sick. So the big challenge that we face here is we need to figure out how we can kill the cancer cells without actually hurting the healthy cells, because the cancer cells originate from our bodies. So they're actually very similar to all of the rest of the cells in our body. So things that might be good at killing cancer cells also might be really damaging to our healthy cells. And so this is a big challenge when people are trying to find ways to treat cancer. Because a lot of cancer treatments can make a person also make them sick. So as I said, as a biomedical engineer, we try to use what we know about biology and medicine to kind of engineer new treatments. So I want to talk a little bit about what we do know about cancer cells and about DNA. So as I said, cancer cells are pretty similar to other healthy cells in our body, but one difference that some cancer cells have is that they have a lot of extra copies of what's called a gene. So you may have heard of genes in biology classes, but it's a piece of the cell's genetic material, the DNA inside the cell. And in cancer cells, this pink gene, for example, that I've shown here, might show up a lot of times in that cell, whereas in a healthy cell, that pink gene might show up only a couple times. And so that's one big difference that we can take advantage of when we're trying to think about how we can treat cancer cells without hurting the healthy cells. So a couple other things that we know about DNA that we can use to design our experiments. So DNA has a special chemistry, and that allows it to stick really tightly to a specific gene and kind of ignore other genes. So what I've shown here is a little piece of pink DNA, which matches up with this pink gene. So when those two come into contact, they'll stick together and stay stuck together. But if this piece of pink DNA saw any of the other genes here, it would kind of ignore it and it wouldn't stick to it. Another thing we know is that DNA, having extra little pieces of DNA sticking to the cell's own DNA is very toxic to the cell. So what I mean by that is if this pink piece did come in and it came into the cell and everywhere you see a pink circle, it stuck there. Ultimately, that would end up being toxic to the cell. The cell would get sick and eventually it would die. So we're trying to see if we can use that to kill cancer cells and have it be less toxic to cells that don't have a lot of this pink cancer gene. So based on all of that information that it just described, we formulated our hypothesis. So my hypothesis, as an if-then statement, is if we give cancer cells pieces of DNA that match their specific genes that only cancer cells have, then the cancer cells will become sick and they'll die. So I'm going to show you an experimental design and some results that will tell us whether or not our hypothesis is supported. So before we can just kind of go in and see, oh, is this DNA going to kill the cancer cells, we have to think very carefully about how we're going to design the experiment so that we know how to tell if our results are working or not. So we're able to grow actual real human cancer cells in the lab outside of a body. We grow them in a little flat, kind of like I've shown here, like a little petri dish. And we can then do different things to these cells, including give them the small piece of DNA that we think will effectively kill the cancer cells. So another thing that I think is kind of interesting component of this experiment is that if we just put that piece of DNA on top of the cells, it won't get inside the cell. And if it doesn't get inside the cell, it won't have a chance to stick to the cell's genes and kill it. So it's really important that the DNA gets inside. In order to do that, we use something called a polymer. So a polymer is a long, stringy piece of material, and I like to think about it as like a piece of spaghetti. So what we do is we take the DNA that we want to give to the cells, and we mix it in with like a big bowl of spaghetti. So when the cell sees it, it says, that looks delicious. I want to eat that. Eats a big plate of spaghetti. And in the process, the DNA that we're trying to put in there gets inside the cells so that it can have its effect. So once we do this treatment, we just have to wait a little while, wait for the DNA to get in there, for the cells to eat that spaghetti mixture, and then wait to see if the cells are starting to get sick over time. So for any experiment, we have to have a way to measure what the effect is. And fortunately, just like you and I, cells have symptoms when they're sick. But unlike us, you can't use a thermometer or check the heartbeat of a cell. So we have to have other ways to look at their symptoms to see if they're sick. And we use lasers in a method that's called flow cytometry. So flow cytometry, I have a diagram depicted here. It's a big machine that takes cells and lines them up single file, one by one. The cells flow through and they pass by this laser beam. And then there's a little detector on the other side that can tell how much light is passing through. So in the data that I am going to show next, the more light that comes through the cells, the sicker they are. So that's how we're going to tell whether or not our treatment is working. So here I
We
have,
have a question, Melanie.
oh yeah,
Uh,
go ahead.
So does the cancer cell have to be marked before the DNA is put into the cell?
Does the cancer cell have to be marked? Um, I guess can you explain that a little bit more?
Yeah, sure. Adam, could you type your little clarification? Do you mean marked with like the dye?
So we do, I'll wait and see if Adam types a little bit more, but we do have to know that the cancer cell is what we think it is and that it is a cancer cell. So there are ways that you can mark cells to make sure they are the correct type of cell. So I don't know if that's exactly answers the question that you're getting at. But yes, we have ways to identify whether a cell is cancerous or not.
I'm gonna go ahead and unmute Adam really quick just to see if you want to ask
Sure.
a
Yeah,
follow-up
that'd be
question.
great. Yeah, thank you.
Okay, Adam, if you'd like to ask your question.
Right. Yes. I see what you're so we don't do anything ourselves to the cancer cells to mark them as having that trait. Um, but we are able to I'm going to come back over to this section of the slide again. So we're able to kind of read the genes and the sequences of the genes inside the cells. Um, and so we know whether or not that cancer cell has, for example, this pink gene here. And we don't do anything to specifically mark the cell, but we can, if we had a group of cells that we thought were cancer cells, we have ways of reading that gene to know whether or not that's that trait, that gene that is going to be specific for our treatment. Does that answer the question? Awesome. Yeah, thanks for your question. It's a good one.
And we have one more question. I'm just
Okay.
going to unmute Rishi.
That's a great question. Yeah. So one thing it sounds like you've learned in other classes that cancer cells do is they multiply very rapidly. And so this is what allows them to kind of take over other healthy cells in our body. And so a lot of times this gene that I'm showing that has a lot of copies in the cancer cell, that pig gene there might be involved in what makes the cells multiply very rapidly. And so we're kind of, I think, using the method you're proposing in a way because we're saying, okay, the cancer cells, we know they multiply really fast. And the reason they multiply very fast is because they have a lot of this gene. And so, therefore, if we kind of use our technique that is specific to that gene, um, then we can target really only the cancer cells, the cells that are dividing really quickly. So that's kind of one way that this approach fits in with what you're describing. And then there are other approaches to treating cancer that do kind of exactly what you're suggesting, which is look at the different parts of the cell that are involved in dividing and making it divide quickly and making a drug that would target that process. Yeah, so that's definitely good. So those are all like it's another example of a way in which the cancer cell is different from the normal healthy cell that we can take advantage of. Because as I said in the beginning, they are similar, but there are some specific differences, and that's what we need to target in order to develop a treatment. So that's a great example of another one. Yeah, thank you.
So we have a few more questions coming up, but we might continue with the presentation and then we can ask more questions when we get to the breakout rooms. That'll be a really great time for everyone to ask their questions.
Great. Okay. Let's see. So I think I was just kind of starting to talk about the results that we have here. So in order to kind of interpret what you're seeing on the slide here, I'll just describe the type of graph that I'm showing. It's called a histogram, and basically what that means is it's a graph of counting. So it counts how many cells had a certain amount of light passing through. So if you recall going back here, as I said, the more light, the sicker the cell. So along this x-axis here, as you move from the left to the right, over on this end, this will be more light, meaning sicker cells, or as I labeled here, dying cells. Whereas over far on this end, over here, these would be healthier cells. So kind of this whole big gray lump here is a counting of how many cells are sick or dying. And this whole lump over here is a count of how many cells are still healthy. So that's how you kind of interpret this graph here. And I drew a dotted line just to make it a little bit more clear that anything over on this side of that dotted line is a sick cell, and anything over on this side is a healthy cell, and the same in this graph over here as well. So that's how we look at this data. And I also want to point out that I have two graphs here. And so this is a time to point out that it's always important to remember controls when you're setting up a science experiment. So if probably many of you have set up an experiment for a science fair, a science class, and this is definitely an important part of it, because without a control, we might get a result, but not really know if we can kind of trust what it's telling us. So in my case, the control that I used was a different piece of DNA that's shown in gray here that doesn't match the pink cancer gene. So this graph shows my control treatment. And this graph over here, as I described before, has that piece of DNA that matches the cancer gene. So we would think if you have that non-specific interaction, that it's not going to be as toxic to the cells. And as you can see here, there are definitely a lot fewer sick cells or dying cells when we give this kind of random piece of DNA that doesn't interact with the cancer gene, compared to when we give the cancer specific DNA. There's a lot more sick cells here. So I'll move on and talk about some of the conclusions that I drew from this experiment. And the first two things I listed are kind of the most direct conclusions that we can make, which is the cancer specific DNA killed some cells, I guess not all, but definitely some. And the non-specific DNA, the control group, had less of an effect. So some of the kind of further things we can perhaps conclude or guess from this is that probably the DNA is getting into the cells. So our spaghetti trick worked. So the DNA is in there, so it can have an effect. We can also kind of conclude that the toxicity of the DNA, or its ability to kill the cell, does seem to depend on whether or not it matches the cancer gene. And then finally, I definitely will note that not all of the cells were killed with this treatment. If you look back at the graph over here, there's still plenty of the cancer cells that were healthy after the treatment. So this is something that, if we were trying to develop this into something that could help patients, we would need to work on that element. But it's definitely a promising result that we could maybe use this in the future. And speaking of the future, so some of the next steps that I came up with following this experiment that I would want to continue to learn more about would be one thing that I thought would be really important is to give that same piece of DNA to a non-cancerous cell. So just to a normal healthy cell that has a lot less of this pink gene. And hopefully we would see that there's those cells are not as affected and don't die as a result of that DNA piece. Another thing that I would like to try and people are working on is to use cells that are actually grown in 3 dimensions. So as I mentioned in the design over here, this little plate is basically means that the cells are kind of growing in a flat sheet, like a piece of paper thickness. And so it doesn't really replicate what our bodies are actually like. Here's a drawing of a liver, if anyone can recognize this organ. But just as an example of, your body is a 3-dimensional structure, cells are all stacked in on one another. So if we can try this treatment on cells that are similarly stacked in 3 dimensions, it would be more similar to how this would actually potentially work in the body. So these are things that people are working on now. They're things that if you're interested in this field, you could work on. And with that, I guess I'll wrap up the presentation and thank you all for your attention and questions. And I will take any other questions if you have them.
Hi, everybody. Our next speaker will be Nick Fisk. He is a computational biologist, and his talk will also be a little bit about cancer, but it'll be looking at it from the perspective of evolution, which is maybe not a way that any of you have considered it before. So let's all give him our attention and enjoy his great talk.
Yeah, thank you, Matt. I just want to introduce myself. I am a PhD student here at Yale in computational biology and bioinformatics. And what that means is that I use math, computers, and evolution to understand and treat cancer and other sorts of diseases. And there's a couple things I want to stress kind of up front here, which is being a scientist is just a job, right? So you can still be a creative person, you can still be an athletic person, you still like sports and stuff, and still be a scientist. It's a job, right? So I like to write poetry and I like to wrestle, and that's all okay. And likewise, there's lots of different kinds of science, right? So I'm a computational biologist, so I don't run experiments. I do stuff on the computer, and that's because my hands don't work very well, right? But that's okay because there's lots of sorts of science that don't require me to use my hands, like people who run experiments. And so you might be wondering a little bit, like, who's this character? Does anyone know the character I have on the screen? You put it in chat. Who is it? Who is it? It's the Lorax. Yeah, the Lorax. And so you might be asking yourselves, like, why is the Lorax there? And I think it's kind of a funny story because my labs and my lab mates and my classmates, they all call me the Lorax because I speak for the trees, right? But not the sorts of trees you'll find in the forest, but instead I speak for evolutionary trees. And so what do I mean by evolution? Well, evolution just generally means change over time. But in the context of biology, we usually talk about evolution by natural selection, or like survival of the fittest, right? And this phrase comes from this idea that organisms that are more successful at having offspring, at replicating, will pass on traits that made them successful to the next generation, right? So if you have something like a giraffe and there's lots of tall trees, the giraffes with the longer necks will be able to reach those leaves, eat more food, and then have more offspring. But that also means if you turn that thinking around a little bit, you can also trace back the relationship between all life on Earth to sort of single points. So all life on Earth is related. So what I have here is something we call a phylogenetic tree or an evolutionary tree, and this depicts the relationship between some animals, right? And so I think it's all pretty sensible to everybody that humans and mice are more related to each other. Oh, sorry, we call this a tree because if this is a tree here, if we turn it sideways, it looks a lot like a tree, right? So it has branches. We even use similar language. We call these we call the sticks on the tree a branch. We call the very end of the tree a root, just like real trees. And we even call the organisms at the end of the tree, we call them leaves. Sometimes we'll call them taxa. And so this represents the relationships, how these different things are related. I think it's pretty sensible and makes a lot of sense to think that humans and mice are more closely related to each other than we are to like worms, right? So we form a group. And I also think it makes a lot of sense to think that like humans, mice, and chickens are all a little bit more closely related to each other than we are flies, right? So these sort of the closeness on the trees represent a closeness of your evolutionary relationship. I think I just saw in a chat that someone said that we are related to monkeys, and yes, monkeys would also be here on this tree somewhere. It would be somewhere in between mice and humans, right? So this is only some animals. It's not all animals. It turns out there's lots of animals. So the way we actually build these trees is that we do some mathematics, a lot of times DNA, to represent how these organisms are related to each other. And so why is evolution important? Why do we care about it? Well, for one, it's kind of the framework that we use to answer questions about the history of life, right? For instance, we used to think that dinosaurs were just kind of big lizards, right? But using evolutionary methods, we actually came to realize that dinosaurs were a lot more like this. They were a lot more like birds than we thought. And indeed, scientifically speaking, birds are dinosaurs. If the label dinosaur has any meaning, you have to count birds amongst them, which is really kind of cool. But let's say you don't care about dinosaurs because they're extinct. Well, we can also use it to understand living things. So here's a quick quiz for you guys. You can put it in, put your answer in the chat. But here are 4 birds: a kestrel, a falcon, a hawk, and a parrot. Which of these birds is not like the other? Go ahead and put it in the chat. D. I've seen a lot of D's. Seeing a lot of D's. All right. Well, I'm gonna, commit your answer, but I'm gonna give you the answer now. And the answer is the hawk. C. What? That's wild, right? But as it turns out, we used to think that kestrels and falcons were more closely related to hawks, eagles, vultures, and owls, right? What we think of as the birds of prey. And that's because their beaks and their talons kind of really look like these other birds, right? But if you look at the DNA and you do the mathematics of evolution, it turns out that kestrels and falcons are actually more closely related to parrots and other sorts of birds like that. And if you wanted to call kestrels, like hawks and eagles and prey, you'd have to include things like toucans. And so, why am I talking about these birds? And it's not because they're important to the story per se, but it's because you have to do the science. You can't just trust what our instinct says, right? People are fallible. You have to actually go through and do the science. Otherwise, just like I used to think, and just like scientists used to think, you would think that parrots were the outsider. In reality, it was the hawk. And what I want to stress is the math and everything we use to figure the stuff about birds, we're going to use it to figure out about the evolution that's happening in you all the time, right? So we're going to focus just on 2 instances of evolution happening in you, one good and one bad. But keep in mind, there's lots of evolution happening into you, not just this stuff. All right. So the first case, the good case, is something called a B cell. And B cells are just one type of your immune system, one type of immune cell. And your immune system generally just fights with your body to fight off foreign intruders, things like viruses and bacteria and things. So this is just one of the members of your immune system, but it's a very important one. So you might want to ask yourself, though, how does your immune system know when something is an intruder versus not? And how does it know which intruder it is? That's useful information, right? And it turns out the answer is it evolves. And so I'm going to walk you through a little bit about how these B cells evolve. So you have this blue sphere, we say is a B cell, and we're going to have this little bacteria, green bacteria here. It's bacteria. And so B cells use these things called B cell receptors to recognize pieces of the bacteria, in this case, this circle that scientists like to call an antigen. You think of an antigen just as some piece of a bad guy, right? Some piece of an intruder that the cells would like to be able to recognize. And so if you know that it's an intruder, you can get rid of it before it has a chance to grow and make you sick. And so what it'll do is this bacteria will get chunked up by different cells, and that little piece will go into the B cell. And if it fits well, then it knows it's a bad guy, and it's going to not let any of this bacteria be around. And so what does this have to do with evolution? Well, it has to do with how this B cell receptor gets shaped, right? How does it know that it needs to be fitting this circle? So it starts off that looking something like this. It has a receptor, but it's just kind of non-specific. And the B cell will divide. And when it divides, it has a bunch of, it'll actually have a couple of mutations. They'll evolve a little bit. It'll have some mutations. It'll be a little different. So you'll end up with this little population of B cells, right? All of its kids, it's split into many cells, and they might have receptors that look like this, right? And then what the immune system is going to do is test to see how well these cells can recognize the antigen, the circle here. And so it tries to bind it, and you can tell that, okay, it fits pretty well in the top and the bottom one, but the middle one wouldn't do so well at fitting the antigen. So the top and the bottom one will actually be allowed, they will be deemed more fit, and more fit this case means how good can you fit this receptor. So the middle one's not allowed to have any offspring, and the top one and the bottom one are. And so it'll do that. And they'll have offspring. But the important thing is how good it fits also determines how many offspring it gets to have. So maybe this top one only gets to have 2, and the bottom one gets to have 3. And then when they have offspring, they change, they mutate themselves just a little bit on purpose to see if they can get even better, right? And sometimes that works out well. Sometimes you get an even better fit, like this on bottom. But sometimes it gets worse because it's just kind of a random process. And so you test them again, and the test gets harder each time because you really want something that can recognize these invaders. So maybe this one, even though it passed last time, it's not good enough anymore. You have to get better. And so the receptor here fits very well, and then this one fits all right, right? And then the other ones don't fit so well. They fail the test. They are not deemed fit. So only these two get to evolve, right? And so they will get to have offspring, and they'll have lots of offspring, right? And so now you will be left with a bunch of cells that can recognize the intruder, right? So anytime that intruder shows up again, you don't have to wait and do this process over. You'll just have a bunch of B cells that can recognize it. Yeah. So, but one of the things that's interesting about this is an evolutionary process, and so we can actually draw an evolutionary tree like we did last time, right? Just like before when we were talking about the evolution of organisms, we actually draw a tree just like we did before. And any B cell that doesn't fit the antigen very well, we call them extinct. They're extinct B cell lineages. So it's just like the normal mathematics of figuring out organisms. And so this is actually the system by which your body learns how to fight off or how to recognize intruders. But no body is perfect. Nobody's perfect, right? And so sometimes your immune system can learn wrong, and it causes things like autoimmune disorders. Something like multiple sclerosis is an autoimmune disorder, and it happens when your immune system doesn't quite learn right, and it attacks some of your own cells. But that's not the instance of cells of evolution going wrong in your body that I want to talk about. I want to talk a little bit about my research, which is in cancer. So it turns out that cancer, just like we heard the other speaker, that cancer is a result of your normal cells evolving. So as you get older, or you do things like smoke or drink, some of your cells get damaged and changes get made to their DNA, just like the B cells, right? But unlike B cells, some of these changes will make them more fit in that they just grow more and grow faster. They'll steal more food and other resources from the normal cells. But unlike B cells, this is kind of a bad accident. You don't want this to happen. And your immune system will actually do a pretty good job at keeping cancer from growing. But every so often, a cancer cell will have a change that makes it hard for the immune system to help or detect it. And so it will grow. So you can see the little precursor lesions are like, they sort of turn into cancer, but the immune system doesn't let it grow. And sometimes, most of the cells that divide, they actually don't turn into cancer, even though they have lots of damage. But every so often, you'll have one that grows into tumors that will maybe even metastasize and cause disease. And so what I do, so just like in the B cells, some of these will fail and some of these will succeed, right? Just like the B cells. And evolution doesn't stop here, though. And this gets to a little bit about what people were talking about the treatment in the last talk, which is cancer can evolve not just to grow and turn into a tumor, but it can also evolve to beat our medicine, right? So we can start with a tumor that's made up of many, many different cells. They're all mutated in slightly different ways, maybe on the order of like 1 billion cells or so. I guess, is that, yeah, like 1 billion cells. And then we treat it, but some of those cells are going to be good at surviving that treatment. Just by chance, they're not going to be susceptible. For instance, maybe they don't use all those gene copies, right? They use different gene copies, so our medicine doesn't quite work on them. And so those cells that survive, they're going to grow. And when the tumor comes back, it's going to now be made up of mostly cells that don't care about your treatment, your therapy, right? And it'll grow back to being, even bigger than it started. And so ultimately, if carcinogenesis, which is just a fancy word for cancer development or cancer growth, is an evolutionary process, it makes sense to treat it like one. And so that's what I do, and that's what our lab does. And so this on the left here is an evolutionary tree, of a lung cancer. And then on the right is the animal evolutionary tree that we were talking about at the very start of this talk, right? And they look really familiar, right? And you can actually look across the top here, and we can build this tree of an individual person's tumor and how it's evolved over time since they were born all the way till they first got cancer, all the way to the point where it metastasized. And we can actually trace the changes that happen during this person's life. And we can also trace the effect of medicine, like on the top row here. And so they're very, very similar. And so what I like to study is I will make these cancer evolutionary trees, and we can understand what's causing the cancers and how they can resist chemotherapy and how maybe we can stop them. And so my research in particular focuses on looking at many of these cancer trees, and I like to call it a cancer forest. And I use the same science that we use to learn about birds to answer questions about how cancer becomes immune to therapy, when does it become immune to therapy, and then when is the best time for us to switch medicines to when, right? And so by looking at lots of these cancer trees and looking at lots of patients, we can actually figure this out. And so I will leave you all on this and take some questions, but I like to think that everything in the world is just trees, and then you can trace the root of everything through the branches and the leaves. So I can take questions now. I guess there was a lot.
Okay, we have a few questions about sort of the immunology portion.
I'll go back
We have one question, which was, how does the body make sure that the B cell with a receptor that doesn't fit the antigen, how does it make sure that it doesn't reproduce?
Right, so these are very special cells, and basically, so one of the things that this actually ties back into cancer, your cells actually know how to self-destruct, right? So one way that they can do this is if they don't bind well, then they don't get the signal to grow, and if they don't get the signal to grow, they're told to self-destruct, right? Yeah, apoptosis, right? That's one such way.
Okay, another question that was also around this area is the evolutionary process truly random, though?
Yeah, so it is random, but random may not mean what you think it means, right? So if you roll, you flip a coin, it's going to come up 50-50, right? But you could also imagine a dice with 6 sides, but 3 of those sides all have 6s, and the other is 1, 2, and 3. And even though that's random, right? You don't know ahead of time when you roll the dice. You definitely would say that it's much more likely that to be a 6 than it is to be a 1, 2, or 3, right? So there are biases in evolution, right? Evolution happens at random, but there are certain things that cause it to mutate in these different sort of ways at different sort of rates. And then of course, natural selection, right, works on that. If something changes, but it actually makes you sick, then you're not gonna survive so well, right? If it makes the cell sick, it's not going to survive and grow. If it makes the cell divide a lot more, it's actually going to grow more than the other cells, right? It is more fit because it can divide.
Okay, let's do one more question since we're a little over time.
Oh.
Couldn't you also get cancer from other things like your own DNA?
So your own DNA is the cause of most cancers. So a lot of people will talk about like if your parents or your grandparents have cancer, then you are at risk for cancer, and that's true too. So some of your cancer risk or your risk of getting cancer is inherited, but it turns out most of it happens from damage in your DNA throughout your life, what we would call somatic mutations. And so slowly over the course of your life, your DNA gets more and more damaged. This is part of the reason we actually get older, is because we don't fix the damage perfectly. And so those actually contribute much more, like maybe on the order of 1,000 to 100,000 times more than most of your germline variants, except for very specific cases. But you can also get it from things like the UV damage, right? Not just getting all over from like UV damage causing skin cancer. And you can also get it from some viruses, right? So there are some viruses that if you get, they happen to, the way they infect you is insert themselves in your DNA. And if they insert themselves in your DNA in a way that makes your cells grow, that can cause a cancer too.
Okay, great.